Hello all, good evening. So I'm going to wait for another 2 minutes. Please let me know if you can see my screen. Okay, is it visible? Okay, so we'll start the session. I welcome you all to the second live interaction session or the week 2 class for Bioinformatics Algorithms and Applications course. So I think I forgot to introduce myself last week. So I'm Lavanya. I'm a research scholar at IIT Madras in Protein Biophysics Lab. So I'll be conducting the live sessions on Fridays between 6 to 8 p.m. So let's get into the session. So this week we'll be doing practice questions on week 2 topics, which are protein structure and function 1 and 2, protein sequence database 1 and 2, and pairwise alignment 1 and 2. These are the lectures. So I'm going to use polls instead of using the chat window because there are too many to keep track of. So if you are new to accessing polls in Google Meet, if you're in mobile, you tap on this symbol and then you'll be directed to activities and polls. In PC directly you'll see this triangle, square and circle symbol which indicates activities into which you can find the polls. All right, so first question for the week. Building blocks of proteins are option A nucleic acid, B lipids, C amino acids, and D glucose. So please use polls. Which among these do you think are the building blocks of protein? Thank you, Anuradha. Can you access the polls? Are you able to see it? So if you're on your PC, click on this activities button. If People have joined through their mobiles. Again, you can tap on the three dots, something like this looks at the bottom right panel, I guess. And you can click into the polls, and one of the polls is active now. Others? So, for those who are joining now, have started with the practice questions for week 2. The first question is launched as a poll. So option A, nucleic acids, B, lipids, C, amino acids, and D, glucose. What are the building blocks of proteins? Okay, I guess people are still joining. All right. So Anuradha is right. So building blocks of proteins are not nucleic acid. That would be for, I mean, nucleic acids are in turn made up of nucleotides and nucleosides, sugar, phosphate, and bases. Right? They are a macromolecule in themselves. Proteins are not made up of lipids. Proteins are not made up of glucose, and therefore they made up of amino acids are the building blocks of proteins. Question 2. Which of the following is a transition? So for people who have joined now, we have started the second week session, and I'm using polls this week to get your answers. First question was building blocks of proteins are nucleic acid, lipids, amino acids, or glucose. And we are at the second question on which of the following is a transition. So, what do you call transition with respect to mutations? Let's take a minute. I hope you're all able to see the polls. Do you have any difficulty in accessing polls? Or is my screen visible? You can also send in your answers in chat box if that's, you find it more comfortable. That's okay. All right. Thank you, Nagamani. Krishna says A to G. Option A. Okay. So we have contenders for two options. Rohini Chinta says T. Okay, two people for A and one for C. But do type in your chat box if you can't access the polls because I'm not knowing now, or unmute yourself and speak if that's possible. Okay, so what is a, since we have two different options, I mean, obviously there is a difference in opinion on what transition is, right? So whenever you have a mutation, and here we are talking about a point mutation. So with respect to nucleic acids, we tend to categorize it as a transition or transversion, right? Transition is when you have purine substituted with purine, or a pyrimidine substituted with another pyrimidine. Yes, so when you have, what are the purines? We had previously discussed, right? We need to maintain the distance between the two strands of DNA constant, or RNA, but since we are talking about T here, we're going to go with DNA. So in between the two strands of a double helix, we tend to or nature has its way of balancing things. Therefore, a purine always pairs with a pyrimidine. Actually, purines are bigger, pairs with a pyrimidine, and the pyrimidine always pairs with purine, right? On any order, they should always pair with the same. Therefore, if you're going to change a purine for purine, the distance is still going to be maintained. Right? It's just one way of visualizing things. Of course, you can always argue that it is a single strand that I'm looking at, and still there's a mutation. Why should I worry about the distance? But this is the concept, right? So when a purine is changed to purine, it is still a mutation, but not that big or that huge a transition when compared to, or mutation when compared to a purine to pyrimidine, or vice versa. The other case is when a pyrimidine gets mutated to purine. So these are transversions, whereas transitions are purine to purine, pyrimidine to pyrimidine. Now, what are purines and pyrimidines? In case you've forgotten, purines A and G. Right, adenine and guanine are purines, while cytosine, thymine, uracil are pyrimidines. So as long as it is A to G, G to A, or C to T, T to C, it is a transition. So what do we have here? A to G is there, G to A is not there, C to T is not there, T to C is not there. Therefore, option A is the right answer, and it's not A to T. I hope that was clear. So A to G is a transition. Question 3. Which of the following statements about the dot plot is correct? A, it is a three-dimensional plot. B, it determines the segments of identity between two sequences. C, it gives the E value. And D, all of the above. So I'm displaying this again. So you find the polls. I'm going to activate the polls for question 3. And here is the question. Of course, you can also see it in the polls, or if you find chat easier, go with chat. Okay, got answers. Kavya, Nagamani, and Anadi say B. All right. Looks like an unanimous answer. Let's just wait for half a minute probably to see if we get some other answer. So dot plot is between two sequences where you compare the alignment, right? So definitely it's not a three-dimensional plot. It determines the segments of identity between two sequences. Yes. Does it give the e-value? No, it's just a more like a visual tool, and you cannot go for large sequences. And the more the length of the sequence, the hard it is for you to keep track of things. Apart from that, you cannot do multiple sequences and measure. But most importantly, you don't get e-value from dot plot, right? And all of the above, of course not. So what's the answer? Option B. It determines the segments of identity between two sequences. Question 4. Gap penalties consist of A. Origination penalty alone. So E values, Krishna has asked about E value. We will be learning that in week 3 or week 4, I guess. That's where it gives the strength of the sequence alignment. So the lower the E value, better is its similarity, percentage similarity. So if something is 100% similar, you'll have a very, very, very low E value, close to zero. Darshana, what is meant by dot plot? Mm, we will be doing a question with dot plot, but to just get it off, I mean, let's say we have a protein sequence P E E K mm, T C, okay, and another. All right, maybe I could do this with DNA. Okay, there are two sequences. I have ACTTGA and AGTTCA. So I'll put a dot whenever I find the same, I'm sorry, um, the same species. Okay, so adenine against adenine. Then when you're aligning G, I have G only here. Next, for T, we'll put two dots here. And then the next T, again two dots here. C, one dot here. A, again one here and one there. This is for better. Okay, I think the visibility was low before. Now suppose I have a continuous sequence A, T, G, C, G, and Nandakumar will get there. That was there in the actual assignment, I guess. And I haven't done that question myself, but I'm planning to do it in now during the session. So ATGCG and ATGCG. Now, for a sequence which is absolutely similar, right? They are 100% similar. How does a dot look? A to A, T to T, G one here and one more here, C only here, and G one here and one more here. So you have 5, 1, 2, 3, 4, 5. Sequence length is 5, and I have dots connecting all these points 1, 2, 3, 4, 5. So if the diagonal is continuous, you know that they are 100% similar. Okay, But here, you see, I'm starting the diagonal here. Immediately, there's no point. There is C and there's G. C in one sequence, G in the other sequence. So it is a dissimilarity. And then third and fourth connect, which means they are similar, which agrees. Again, at fifth position, there is no dot, which means they are dissimilar. Correct? And then sixth position, there is no continuity as such, except for the third and fourth and the initial two uh, sequences. Right? So there's one gap here and another gap here, which is shown by a discontinuous diagonal. Right? So it determines the segments of identity. So this is segmented. And if you had more stretch of amino acids between these two T's, you would have had another huge segment of identical sequences or identity between these sequences. Was this clear? Do you want me to repeat? Darshana and Nandakumar. We'll go about the diagonal. So when there is a shift in something, then your diagonal will switch. Thank you, Ramamurthy. Right. Next, question 4. So I think you're all comfortable with the chat rather than polls. So I'm going to skip switching on polls. So it's better that you keep using the chat box. Gap penalties consist of option A, origination penalty alone. Option B, penalty for changing purine to pyrimidine. Option C, origination and length penalties. And option D, penalty for changing purine to purine. So what is this? Purine to pyrimidine is a transversion. Purine to purine is a transition. Okay. I've gotten a lot of C's. Nice. So what are we talking about in gap penalties? Whenever you have so there are two gaps here. So this is an origination of a gap, and here you have an origination of a gap and a continuation, right? So you will have penalty for both starting and continuing. Generally, the penalty for lengthening the gap will be smaller than or lesser than the penalty that leads to origination of a gap, right? So you are counting on the more you start a gap, the more is the penalty on the alignment. So it has both origination as well as length. Why does this matter? Because this is more costly than having a gap which is continuous of three places, right? So answer C is right, as given by a lot of people. I'm happy. Okay, moving on to question 5. Which of the following amino acids does not contain chiral alpha carbon atom? Okay, I think it's Sai Shri. Okay, good, nice. So everybody is clear that all other amino acids have an R group, whereas glycine has the R as H, and therefore it's the only amino acid that does not have a chiral alpha carbon, right? I hope everybody knows what. Right, so for alpha carbon to be chiral, all the four groups have to be different, but in glycine you have H here, therefore H and H are same. It's not chiral. Question C. How many subunits are present in hemoglobin? Option A, 2 alpha 2 beta, option B 1 alpha 1 beta, option C 3 alpha 3 beta, and option D 2 alpha and 1 beta. So although this may not be practically right, or uh, so hemoglobin is a, most of you have gotten it right, that's great. It's a heterotetramer, right? So when you say tetra, you can there only get the option, because all the other options give you 2, 6 and 3 units in total. Therefore it is A. That's a lame way to do things, but you know that there are 2 alphas and 2 betas in hemoglobin, therefore that's the answer. A is the right option. Okay, question 7. The alignment score for the following alignment: match score of 1, mismatch score of -1, and gap penalty is 0. So there's no, first of all, there's no origination and lengthening to worry about here, because each gap is just of length 1. So what is it? You have 1 plus dash 3, 6, 7. I'm just giving you time to work it out. So first one is a match, second one is a gap, third is a mismatch, right? Fourth is a gap again, fifth is a mismatch, and sixth is a, oops, third is a match, right? I made a mistake there. And fourth is a mismatch, fifth is a gap, sixth is a mismatch, seventh is a match. So what do you have? Somebody got -1 and 2. Rework your answers. I think Kavya changed her answer. Good. Nabajit Mondal, look into your answer again. So first is 1, mismatch is -1, gap is 0, match is 1, gap is 0, mismatch is -1, match is 1. So you end up with 1 as the answer. Yes, thanks Navajit. I'm sorry, Anadi, or I hope I'm pronouncing your name right. I think you should write to the supports group, or put it in the ask a question tab in the course. Maybe they'll be able to help you with it. I'm not the course TA, therefore I cannot tell you uh, whether it will be taken for credits. Dashana, which question? The alignment question? Okay. So how do you calculate alignment scores? Here is a question of A, T, A, G, A, T, A. Okay, and then there's the second sequence which has two gaps. Okay, and you're going to see how similar these both are. Now to start with, what are the places that are absolutely same? So which nucleotides are exactly same in both sequences? So those are matches. Okay, so for each match we need to score this alignment, right? You can say they are similar, dissimilar, and walk away. but you need to know between sequences how much the similarity is, or how much is the dissimilarity. You need to be able to quantify. That's where we bring in scoring. So those that are absolutely similar are matches, and for each match the score is +1. So you get a +3 for this. So while I hold on to that, I want to know what are the mismatches. So G to A is a mismatch, and T to C is again a mismatch. So what is the score for it? -2. So -2 into 2. So I have one more, and there are gaps. So against T there is no other sequence. Ideally this is just A, and then you found out there are two gaps between here, and you're aligning with a 7 base sequence, right? There are two gaps, but since the gap score is just 0, sometimes you also have a negative score for gaps, so it could be plus or minus or whatever you want to call it, 0. So therefore your answer is 3 minus 2, 1. Was that clear, Darshana? All right, question 8. Which of the following amino acids have the least occurrences in protein sequences? Is it the pair alanine and glycine, or glutamine or glutamic acid, sorry, and cysteine, proline and lysine, or cysteine and tryptophan? Okay, a lot of D's. Muhammad feels A. Okay, let's hold on to A and D. Uh, yes, Neha, will you be able to wait till the end? We can definitely go over what happened. But for now, please answer the question on the slide, question 8. Good. So we are starting with most occurring towards the least. If you go, it was something like LAGBS, going on towards ETDRKPNFQYMHWC. So I'm writing the single letter codes of occurrence. I missed some here. I think it's proline. Yeah. Oh, there's already proline. You can figure it out with this general idea. So lysine, leucine, alanine are the most probable. But here we are worried about least, right? So you should go from this end. Cysteine, then tryptophan, histidine, methionine, and that way. Therefore, your answer is cysteine and tryptophan. Glutamic acid is wrong because you have it here, right? Glycine is again wrong because you have it here. Alanine again wrong. And compared to proline and lysine, cysteine and tryptophans are more towards this end. Therefore, go with D. Okay. Question 9. Major aspects of UniProt is or are: option A, high level of annotation; option B, less redundancy; option C, high level of integration of data; and D, all of the above. I think this particular information was repeated multiple times during the lectures, and yes, you're all right. It is has both high level of annotation, redundancy, integration of data. So therefore, it is all of the above. It will have, yes, but I think what you mean here is that since it is connected to a lot of, it is widely integrated, it has more redundancy, but still you will be able to tweak it to get your the amount of data that it has over wide range of organisms and wide range of how do you say, the way it has been annotated so much is that it is not redundantly going over the same sequence as a, when you see look at it as a database as a whole. Okay, I mean it's one of the very well networked or connected with very good annotation and integration of the data. And don't think of it as, since it is integrated in so many things, it is redundantly giving you the same information, but compared to, it is not giving it to you as separate entries, right? All of it is collected to a single uh, point, if you think about it. Okay, so this is just to make you think. So we saw transition is A to G, or G to A, or C to T, or Kavya, think again, T to C. Okay, so what about these are all transitions. So what about transversion? So whenever I pair A to T or A to C, vice versa, T to A or C to A, G to T, T to G, G to C, C to G. Now think about it. So these are the points you need to worry about now. Is there A to T somewhere? Yes, correct, Krishna. G to C somewhere? Yes. A to C? Yes. T to G? It's here, right? So all of them are transversions, right? Okay. So for I'm going to discuss the assignment for this year, but before that, for people who joined late, we'll go over a very quick uh, recap of what we did till now. Neha, this is for you. So we went over the we are doing the practice questions for week 2. These are the topics, you already know that, and I had expected people to use polls, but that's okay. And the building blocks of proteins are amino acids, obviously, and not lipids, nucleic acids, or glucose. Question 2, which of the following is a transition? So we saw point mutations classified as transitions or transversions, and we are talking about nucleic acids here and not between proteins. Okay, so nucleic acids. So transitions are changes between furin to furin and pyrimidine to pyrimidine. And transversions are when a base changes from furin to pyrimidine or pyrimidine to purine. So here it was transitions, and we saw only one option held true. These were the possibilities: purines to purines and pyrimidines to pyrimidines, which was A. Okay, question 3. Which of the following statements about the dot plot is correct? It determines the segment identities between two sequences. We did a small example to see how the sequences are similar or dissimilar. how it would look if it were similar, and how it would look if there is a mismatch or a gap. I think we didn't do a gap, we can do soon. And four gap penalties consist of origin and length penalties, and not just origin, or it does not worry about transition or transversion. That is for mismatch to worry, or it belongs to the mismatch scores. And gap is to do with only option A and C. Definitely it is option C. Although there might be cases which go with just origination penalty alone, but the ideal way is to have two penalties in gaps, one for origination and one for the lengthening. Okay, and then question 5 was, which of the following amino acids does not contain chiral carbon? So the amino acid that has a hydrogen atom in place of R group in the format for amino acid that we generally use in proteins, that would be your answer, which is glycine. All the others have a side chain or an R group, therefore making the alpha carbon chiral. Question 6 was, how many subunits are present in hemoglobin? 2 alpha and 2 beta, therefore heterotetramer. Option A is the right one. Okay, so for Rambala. Chiral carbon, so whenever you have a carbon connected to four different groups, A, B, C, and D, it will give a mirror image of itself, and those are called chiral molecules. All right, there will be two isomers, and these that is so in the presence of a polarized light, these carbon atoms or molecules, when connected to four different groups, will be able to rotate in clockwise or anti-clockwise direction. So these are called L or D isomers, and these isomers L or D, yeah, so they are called stereoisomers, and that carbon is called chiral carbon. Okay, so amino acids, as we all know, is connected to an carboxylic acid and amine group in the end, and this will get ionized to give. All right, and then since the valence is 4, on one side you have hydrogen, on the other side you have the R group, which is the side chain moiety, or where all the substitution occurs to give different amino acids. So you have 20 different amino acids based on this R group only. So if it's just a H, it is glycine. If it is CH2 or CH3, to be fair, yes, CH3 is the R group, it is alanine. CH2, CH3, it is valine, and so on. There are certain aromatic amino acids which have aromatic side chain and stuff, right? So these are the, yes, optical isomers. Therefore, glycine is the one amino acid that cannot give rise to isomers, and because it does not have a chiral carbon, right? And length penalties with an example. Okay, so for Krishna, suppose I have a gap penalty of -3 for origination, okay, and -1 for length, okay, and match score of 2, mismatch score of -1. So remember this, I'm putting arrowheads, so oops, this is minus, this is +2 and -1. Okay, let's take a sequence now. Even this holds good, right? I'll just change the color so you don't get confused. So C to C is a match, A to A is a match, T to T is a match, G to G is a match, and T to T is another match. There are no mismatches, so I find this boring, and I don't know why I'm unable to. Okay, let's say this is A, so this is a mismatch here, right? So now I'm going to score this alignment. How many matches do I have? Matches 4, mismatch 1, gaps are 2, right? You have two gaps here of length 1 and 2. Now how will I do this? So for 4 I will multiply 4 into match score is 2, 4 into 2 is 8. Mismatch 1 into minus 1 is minus 1. Now for gaps here, the first gap I'll give it a minus 3, and for the second gap there is a minus 3 plus, you're just extending this gap and you're not starting or introducing a gap here. So the length score will apply minus 1. So how much do you have? Minus 3 minus 3 6, minus 1 7. So you have 8 minus 1, then minus 7. When you add these up, it will be 0. I didn't expect to end with 0, but 0 is what we have. Was that clear, Krishna? For yeah, okay, so I'm going to repeat the same thing. C T A T G C G T. Okay, and C gap A G gap gap T. Now let's do it stepwise. So C to C is a match, right? So I'm going to give it a score of 2 plus T, and there's a gap. So this is the first gap. I'm giving it minus 3, and then A to A is a match 2. T to A is a mismatch. Therefore, I'm giving it minus 1. Clear? I don't know who spoke. And G to G is a match 2 plus C to G is a first, again a new gap is getting introduced, therefore I'm giving it minus 3 for origination of the gap. Okay, and then G to another gap, there is one more gap, but it's a gap in progress, right? It is still a single gap but with two places, 2 points. So if this gap were 3 places, no, I'll give minus 3 only for the first point, first gap, and then it will be minus 1 and minus 1 because that's what I'm using to lengthen the gap, right? So C2 gap is a minus 3, and G for the next gap is minus 1 plus T to T is a match, so I'm getting 2. Now let's see. 2 minus 3 minus 1 plus 2 is plus 1, minus 1 is 0. Again you have 2 minus 3 minus 1 2, which is 4, and minus 4 0. Do you get it? Kiran, I am just giving random scores here just for the sake of showing an example. And note here, the only thing you need to observe here is that the penalty I'm giving for origination is higher than the penalty I give for lengthening. Okay. Otherwise, it's just a random score. If I had given minus 2, this you could have ended up with a plus 2 here. But since I gave a big penalty for minus 3, I'm getting a 0 answer. All right. Okay. So that was question 5. 6 also was done. 7 we have done another alignment with match, mismatch, and gap scores. And here for gap there's just a 0. Therefore, not much damage done. And I hope you all understood this question. Especially after doing that, now this should seem like a cakewalk. 3 minus 2 is 1. Question 8 was, which of the following amino acids have least occurrence? Generally, cysteines and tryptophans are all histidines, methionines, even tyrosines are less in occurrence. Okay, compared to the amino acids towards the front. I mean, you cannot remember all this sequences, but it's safe to just know that tryptophans or cysteines are rare. So hemoglobin for Ambalas, or I hope I am pronouncing your name right. So, or R Ambala, I don't know. Okay, so hemoglobin is a tetramer, okay, which means it has 4 subunits, and it's a heterotetramer, which means it is not made, all 4 are not same. Okay, and it is made up of two homodimers. It has two alpha and two beta. Okay, and this is not common for all protein structures. It is just for hemoglobin. I hope that is where you had your question. Generally, proteins will have the primary sequence where you just have a sequence of amino acids followed by secondary structure, which is helix or beta sheet. And then you have the tertiary structure where it actually gets a proper fold and you know how the amino acids are oriented. And sometimes different subunits of protein, the protein, in order to be functional, can have multiple subunits which have a structure of their own. Each of these subunit will have a proper structure of its own. Okay, but they have to come together in order to be completely functional for the protein to do whatever it has to do. So in this case, it's a heterotetramer with two alpha and two beta. How do we decide where to put the origination and gap? So whenever you have a single gap, it will be the origin penalty. Whenever you have two, it will have one origin and one length. Three gaps, origin, length, length. But even if there is one match or mismatch, whenever you start a gap, you have to put the origin for the first place. Before that, you might have had another gaps. Again, you will put the use the origination of gap penalty whenever a gap is begun because you're starting something. And then it is continued, you'll use length. Okay. Then major aspects of Uniprot was already discussed. This was just to test your. I just don't want you to jump into one answer thinking it's an MCQ, so just give a lot. All right. So going into week two assignment. So I have polls for this also. You want it or chat is okay? I think we'll go with chat. Drop in your options. You've already submitted this assignment, I believe. In case you haven't, this is your chance to learn, but I don't think you'll be able to help yourself with assignment scores. Rank the following amino acids by increasing polarity. So start with the least polar to most polar, or most hydrophobic to least hydrophobic. I'm seeing two different answers. Nice. I mean, have you all attempted this assignment? I think the deadline was 8th. Okay. And generally, we have the live sessions such that I don't think so. You can contact the team, but I don't think they would allow you to submit it beyond the time deadline. So keep track of those. Each week, I think around today is 10th, right? So Wednesday. So by Wednesday, I think every week you have to submit your assignment. Otherwise, it expires. This is in case you're trying to get yourself certified for the exam or for this course, or you went planning on attempting the exam. So if it's fine if you have, I think there's a minimum of 8 or 10 assignments to do. I can get you that information maybe next class, but please be careful to submit. Until 12 a.m. is also fine, but before Wednesday night, you should submit in your assignments. Yeah, actually, yes. And the first two assignments had the same submission. That is generally the case. From I don't know who's talking actually, but I really don't. Neha, okay. I don't think so, Neha. You can post it in the ask forum, ask a question forum, or I can get you a mail ID where you can ask your queries, but I don't think it will be graded because the answers will be put out already. I think, right? It's all right. You still have the other weeks to work on. I'm just sad that generally before the deadline we'll have the live sessions scheduled. So the sessions will actually help you to do the assignments, but this year it's a little skewed. So after the assignment is deadline is over, we are having the sessions. I don't think we can do anything about it, but you can learn. So that's one plus. So rank the following amino acids by increasing polarity. So which amino acids are more polar and which are less polar? So this is the main chain is not changing. It's the R group that varies between amino acids, right? This was displayed in your lecture. I'm just writing it down here so you have something to help you decide. Some of you have given the right answer, others haven't, or some are still thinking. So this will help you think more. Now think about this. What options do you have? Aspartic acid, right? So there's one here. Serine. What does serine have? Okay, I have something to help you guys here. So I put it in my. Oops, oops, oops. Just bear with me for a minute. Okay. Yeah, I agree. It's a little difficult to memorize the structures, but you can always think about it as glycine. See, the first five are very easy. I mean, this is a nice picture. Maybe you can look at it this way. So this is H, CH3, then it becomes 3, CH, CH3, CH3. Then you add one more CH2 to this, and you get leucine. And it's the same four carbon chain, but you just go with an isomer. So it has shifted to this carbon being attached to C alpha, and the others follow suit. Okay, isoleucine. And I mean, this is just a way to remember. Don't think this carbon really went and did there. And then the same four carbon structure follows with sulfur here. So this is one of the sulfur containing methionine, and cysteine is another sulfur. This is how Sir had classified. And then you have the aromatics, phenylalanine, tryptophan, proline. Okay, so these are all non-polar side chains. For polar, you have serine because of the OH here. Threonine, again because of the OH, you have CH2OH and CH2, CH3, and an OH. Then CH2SH. Serine and cysteine are very, very similar. It's just that O is replaced with S. And then you have tyrosine, asparagine, glutamine. And both of these, remember, have CONH2, which makes them amide. They fall in the first group. These fellows are the most polar. Okay. And then you have acids. So what are the acids? Aspartic and glutamic acid or glutamate or aspartate. Alright, these are the second winners. And followed by alcohol. What all alcohols you have? One, two, three. So these are the third group. Alright. Followed by amines. What do they have? NH2. Arginine, histidine, and lysine. Yeah, I think I've covered. Right? These are all non-polar. Alkanes, where you have all these guys falling into GAVL. Right? Okay. So according to this, you have one acid, and serine is a OH group. So there's one alcohol, and threonine is another alcohol, but it has a bigger carbon length. There's one more CH3, right? Which makes it less polar compared to serine. So threonine is less polar compared to serine, which is less polar compared to, right? So which one does this resemble? Option C. Yes, clear? All right. One PAM means one accepted point mutation per. Okay, okay. So many A's. Neither answer. That's right. It is actually 100. Okay, option A. It's nothing, Nagamani. It's just that one PAM matrix is how many mutation per. It is one substitution per 100 residues. That is called as a one PAM unit, which stands for point accepted mutation. We will anyway go over this on a lot more detail in next week. Yes, PAM 250 and 1000 also we'll be doing. I mean, I'll be explaining. I think there'll be questions on it in next week. And what is the difference? When should you use what? Right? Okay, choose the amino acid with the side chain imino group. That's right. We'll give another minute for others to think about it. Okay. So we just saw, right? Serine has, or instead of seeing anything, you know for a fact that proline has an imino group. Ashera, we just saw the structures, right? This is lysine, and what were the other options? Serine, proline, methionine, and then there's serine here, and proline. Where's proline? Oh my god. Yeah, here's, yes. This is the imino group because of C being attached to NH2, and it is actually like this. So it is bound on both sides and in connection with C. Therefore, this is the amino acid and not the others. And then there was serine, threonine, something, some other option, but you know methionine. Obviously, you know methionine is not the amino acid, somewhere here, right? Okay. The presence of a gap in an alignment is due to option A, insertions only, option B, deletions only, option C, both insertions and deletions, option D, none of the above. Good to see you all react. So if I have two sequences like this, they are same in so many ways, but to a plain eye, it doesn't feel right. So TCATCGTC and TCGATGTC. So you have similarity here, and dissimilarity in this region, right? Now, how can you think of this? TCATC, GTC, GTC, there's a gap, TA, and you have a G here. So if I give a gap here and put CT, so you have 3 plus 2 plus 2, 7 compared to 5 there, right? Gaps are given both by insertion or deletion, depends on how you think about it, but both insertion as well as deletion will give rise to gaps. So for the following alignment, match score 2, mismatch score -1, and gap -2. Let me put it here because I think you will miss it on the other side. Gap of -2, what's the net score? Most of you have done the assignment already, therefore there are two matches, then mismatches 3, right? And gap is 1. So you have a match score of 4, mismatches -3, gap is -2. So that will give you -1, right? Yeah, somebody had asked for this, right? How do you get below the diagonal, above the diagonal? Let's see what happens. When constructing a dot plot using leader on the x-axis, I'm just reading it. It's a amino acid sequence. Continuous dots more than three are observed where y-axis ADERFE. Okay, ADERFE. So I had planned on working this out here because I didn't feel like drawing a dot plot while I made the slides. So I'm going to put, and the other one was ADERFE, right? So we're going to use only this space here. All right, L, there's no L. E, you have one, you have two. Okay, I'm supposed to be putting dots, right? As the name goes. And A, I have A here and no other A. And then there's D here, nowhere else. And then there's E. E is here and here. R, we have R only here, right? So where is this? And what was the question? When constructing a dot plot using the following sequence, continuous dots more than three are observed. And here is your diagonal. Don't mind the bad line. Do you know? Okay, so it's below the diagonal, right? Next, structural proteins are option A, globular proteins, option B, fibrous proteins, option C, alpha helical membrane proteins, and option D, beta barrel membrane proteins. No alternatives. Okay, yes. So it is the fibrous proteins that generally give structure. Alpha helical and beta barrel membrane proteins often function as receptors or transport proteins at the membrane level, or receptors, or those that interact with other cells or other external environment. Globular proteins are have multitude of functions, but generally the structural proteins which are referred to in this lecture course, or how they mean it as in giving strength to cells, mostly fall under this fibrous protein categories. Okay, an example of blood clotting protein is option A, immunoglobulin, option B, monelin, option C, insulin, and option D, thrombin. Burst of answer. Immunoglobulin is the one that you have in antibodies, right? Monelin was mentioned as a sweetener, and insulin is the enzyme that helps control blood sugar levels, and thrombin is a enzyme which is involved in the clotting. Oh, I'm sorry, insulin is a, yes, yeah, it's the hormone. No, pancreas, yes, yes, sorry, yes, hormone which controls blood sugar levels. This is involved in the clotting. process. That's right. Thanks for noting that it's part of the endocrine system, right? Endocrine process. Yes. So fixed, right? As everybody said, D. Option. I'm sorry. Question 19. Enzyme number 3212 belongs to. Of course, you are not expected to remember the classification of everything. Just go with the first number. Oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligase. So option 3 is, I mean, 3.2.1.2 should belong to category hydrolases. So since we have time, and amongst these 10 questions, do you have any problem with anything? Because when I went over first 10 questions, I realized a lot of you had doubts or questions in it. So if there's something like that similarly, please let me know. I'm just going over for you to ask. I think we went over all of these in detail, so shouldn't be a problem. Okay, so but let's do this small exercise of getting past a sequence from Uniprot. So I'm planning to do this with you as well. Yes, Kiran. So others you can start looking for. I haven't mentioned what lysozyme or human or animal or anything. You can just go explore Uniprot or listen to the question or the explanation I'm going to give for Kiran. Yeah. So these are the list of 20 amino acids, and you know that the whenever you have long side chain of carbon, it is not going to be very happy with water, right? Only when you have some ions or other polar groups like this, water is going to interact with these side chains, but not with an aromatic side chain or where you have a long carbon side chain. So these are things that they don't like water, doesn't like them, so they just tend to huddle together and form hydrophobic interactions. So polar amino acids with polar side chains are hydrophilic generally, and these are hydrophobic, right? Electrically charged again will be hydrophilic, but not as much as these guys, polar side chains. And again, they will be forming their own connections between negatively and positively charged amino acids. Now here we had a sequence, so I don't want to corrupt this chart. So I'm going to leave it clean for you to see. So I'm going to write amide is more polar than acid. I'm using the same series so that you will be able to follow the lecture as well as this with the same alcohol followed by followed by alkane. These are all alkane side chains, right? So to know what groups these are, I'm going to write that also here. Amide is ONH2, acid is COOH, CO minus, alcohol is just OH, amine is NH2, alkane is CCC, or how many ever C's there are with H and stuff. Right, I'll just go with R. So by just viewing at things, asparagine should be the most polar, right? Followed by glutamine, simply because it has an extra carbon length. Okay, the more you're going to add carbons, the more it is going to be non-polar. Correct. And the question that was given was, among rank the following amino acids. There are just three amino acids here. Aspartic acid, I'm sorry, made a mistake here. Yeah, ASN. But the question given to you here is ASP, SER, and THR. Okay, I am going to just color those alone here. There is aspartic acid here. Then serine. And then threonine. Now look at this plot. Followed by acid. Which acid is more polar? Aspartic. Followed by glutamic acid. So asparagine, glutamine, aspartic, glutamic acids. Alright. Then comes alcohol. So we're going to have serine followed by threonine followed by tyrosine, I hope. But this covers your question. Serine, threonine, and aspartic acid. So 1, 2, and 3. And they had asked increasing polarity. So this is least, better, best of these three. Therefore, it will be threonine followed by serine followed by aspartic acid, and not this one. Option A and option C are very similar in the order of placing. Oh, sorry. Aspartic, serine, yeah. So this is 1, 2, 3. This is 3 to 1, right? But the only problem is rank the following in increasing polarity. Here it is decreasing polarity. Therefore, you have C as the right answer. Was that clear, Kiran? Okay. Yeah. So just going to look for, so these days it gives you an option for cards or table. I'm going to go with table. So you have mouse, chick, E. coli, rat, human. So I'm going to go with mouse. I hope all of you would have gone over the details. I mean, at least tried how Uniprot feels like. So I can directly go here and click sequence, and here you have download, add, highlight, and everything. So I'm going to click on download. Immediately displays the FASTA format. Right, this together gives you sequence of lysosome from mus musculus, which is mouse. Okay, somebody has given probably human or something. Yeah, nice Nagamani. So we'll be in I don't want to dump so many things before it crops up in the lectures. We will do all alignment, cluster and everything. Multiple sequence alignment as we go with the course. So this was just a small peek into what's going on. Apart from this, if you have anything else to ask, or if you want me to do another example of alignment or dot plot or something, please let me know. Otherwise, I think I'm done for the day. Somebody had asked me about version number and gene index. Yes, Kiran. So I don't know if you saw, but there's a channel which I maintain for the recording for the live sessions that I take, and I think you should get the link by now. I don't know whether it's already displayed or not. I don't think you got the. I'm sorry, but soon you'll have a tab towards your left where you have all the courses, right? Okay, still it's not available. Darshana, it just comes with time. Yeah, as you just have to keep looking at the table maybe 2 times a day or 5 minutes a day, and it kinds of sticks. Maybe after the course is done, you might forget a little, and then again, once you brush up, you'll remember. There's no method to learn everything, but the colors definitely help, I believe. At least this picture looked clean for me, the one that I just used to show, where you have all the side chains highlighted and split into polar, non-polar, and stuff. Yes, Subbalakshmi, I will do that. But yeah, that's right, Kiran. I don't think any link is provided yet, but generally you will have a link coming up here. Until then, if you want to go over the lectures, I don't know how you get it through. Somebody has unmuted. Can you please mute yourself? This is the week 1, maybe I could post it here. Oops. Please mute yourself. I don't know who's speaking. Okay. So I'm going to paste this link in the chat. So I mean, this is just the first week's link, but I'll be posting all the videos in the same channel. Hope this link works. Yeah, it does. All right. Yeah, I was talking about the other question that you had. Somebody had, I don't remember who it was. Somebody asked about the GIN version, the gene index or GI number. I think it's not gene index as you identification number. Simply the series of digits that are assigned consecutively to each sequence. So NC by BI while recording it gives you a GI number. Okay, and it bears no resemblance to the accession number of sequence record. It is shown in the version field of the database record, and version is made of the accession number of the database record. So these are just so accession dot version number, and it says somewhere that GA number has been used for many years by NCBI to track sequences histories in GenBank before they were merged and stuff, I think. So, and other sequence databases it maintains, so you can go over stuff like this, but I'm really not uh, these are details that I'm not very familiar with. I'm honestly telling it to you. I just Google like you all and find the answers. All right. So there's this website, right? Database where you have catalytic site atlas, which sir was explaining to find binding site. See, one good thing about bioinformatics is that it's really user friendly. All you have to do is just explore. I mean, when I was doing this course as a student or during my UG days, I looked into all these databases, and they have improved over the years. And there are details that I had not noticed before, but which I am discovering now. So you can always Google, click on things, and find out. Go over the databases, all this intact, ProSite, catalytic site atlas. I've never used them to be honest. So, but there's nothing stopping you from trying out new things, right? So, what else? I'll be posting this video also into the same link. Sure, Jui. I mean, you don't have to hesitate. Sometimes I won't know, and I would just tell you I don't know. But you can ask whatever you want to ask. Oh, that's not possible. First of all, I'm watching the lectures like you all do, and I think it is actually well paced only. It's not too fast, and more importantly, it's a rerun course, which means these lectures were recorded at least 2 3 years ago, I believe, because most of these references were from 2011, 2013, 14, or 15. So I'm guessing it was probably first run in 2016 or so. Therefore, you can take time. There's always this live session, and the beautiful thing about this semester is that there are 4 different live sessions each week. Generally, there'll just be one or maximum two. and four different people teaching you. So whoever you're comfortable with, you can follow their live sessions, or you can pick and choose which session to attend in a week according to your comfort. I'm not sure about the timings, but there's one on Friday, Saturday, Sunday, and Tuesday or Monday. I'm not sure, but it's there in the announcement. Okay, that's all right. But if it is part of the lecture course, and if sir has taught it, and if you do not understand, you can always ask me, because I go over the lectures before I start the live session. So I should be able to help you with that. In case sometimes the MCQs or questions that we have will not cover all the points. I agree, but you can ask here, and if I know, I'll help you out, or I'll get back to you like this GI and version number. Anything else? Thank you, Julie. Thank you all for attending this session, and hope to meet you for the next live class. Okay. 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 So, is there anybody else like this? I mean, that's a good point. Thank you. Am I pronouncing your name right? Rambala? So, Rambala? Bala. Okay. It's like some Ramya Bala or something. Okay. So, I mean, we could definitely do that. I hope until now you have understood what are the keywords or whatever we have been talking about, right? Until week two, in case there's something that. Okay. 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 I get it. Because I'm finding some students here who are way into computational biology. Even I mean, uh, most of the people, bioinformatics is just a basic course for them, and they are doing much more and asking questions which I'm not so much of a computational biologist myself. uh, the problem with online courses is that there's a mixture of audience, right? Both people who are just getting a feel of the course, beginners as well as undergraduate students, all the way till industrialists and people who are doing research. So that's a very good point. We will take it into, or at least I will take it into consideration when we do live sessions. And I hope I'm explaining all the basics when I start. In case I'm not, please feel free to put a message in the chat, because I am constantly looking at the chat box even while I explain. So I think, yeah, please feel free to ask. I mean, the session has been made 2 hours from 1 hour. Previously, it was 1 hour per week. So this is absolutely for you to familiarize yourself with the concept and how to solve the questions. So do not hesitate. Please feel free, and I'll help you to the extent I can. In case I'm unable to, I'll either put a description or something while I upload the video on YouTube, or I can also extend the video by giving some explanation, and I might put it there in the description. Those who have already attended the live session shouldn't waste time in watching the entire session, right? So I will tell if I'm including something into the video, or I can also, if something can be verbally said by written comments, I can put it there. And there are many beautiful charts, but this I liked for today's session. Yeah, no problem. I'll share the PPT also. I think I could share the link for that as well. Just wait a moment. If anybody is in a hurry, you can leave. I'm just holding on to the session to clear such questions. But so let me post this also. This is the drive link for my PPT. So let me see what opens here. I think only the first week's PPT comes. So I will maybe give the access for the whole folder so that you can get all the contents with the same link. Okay, so whatever we used in the session, I just uploaded without changing so that you can follow with the video and look into the slide. Okay. Is there anything else, or should I change something about the way these sessions are going? I mean, I tried to use polls, but I think that didn't work. Oh, some people have answered later. Okay, great. If there's something else you want me to look into or address in the next session, please feel free to tell. Yes. What are the location of pyridoxal 5-phosphate binding site for myoglobin protein? Okay, so this is a. Where did you get this question from? Or okay, and you couldn't find it in the. But I think you should do this assignment as since it's part of your college stuff. I would leave it to you. In case you're still unable to do, we will discuss it in the next week. Somebody raised a hand. Is that by mistake or? Thank you, Nanda Kumar. You can see myoglobin structure from PDB, and I would recommend you to use catalytic site atlas, although I haven't used it myself, Thank you. All right, then. So, if there's nothing else, thank you very much for attending. Let's meet in the next session next Friday, 6 to 8. Thank you.